All right, you can close this window. All right. So we're on the main study. I've got it highlighted. So Partech is built on kind of around an ANOVA model. It's a really good ANOVA model in that I can take two different variables and basically test them together. Like what effect does, say, the cell type versus the day have on these genes? And that's a huge thing. And a lot of what kills me is, somebody will have a study like this and they'll do like 10 different t-tests. And I just want to scream every time I see that. Every time you do a statistical test, you're generating error, right? You're doing so many statistical tests that, given a list of 50,000 genes, if I have a p-value of less than 0.01, I'm getting 500 genes that are showing up just significant due to random chance. Okay? So I don't want to, basically my deal is I want to do as few statistical tests as possible. Here's what we're going to do. So let's go to, so we're here, go to stat. Okay, under your stats, go to ANOVA. You should see this window. Can everybody see that up there? Do I need to mess with the resolution or anything? No? Okay, good. All right, here's what I want to put in. We basically have two variables in this study. A lot of these variables are kind of like the sorting protocol goes with day, cell type clone, that's pretty much related. So basically what we have is day, so put in day, so highlight day and then go add factor. Next we're going to pick cell type. So this is going to be, remember we're treating all the pluripotent stem cells the same, so we're grouping them together. So add this, add factor. Now here is where I'm going to say, okay, so basically we're going to find genes that change due to basically differentiation state and due to embryonic versus pluripotent state. But now I want to see what genes change due to both these factors together, because that might be relevant for what we're looking at. You don't have a day? You could put in sorting protocol, I guess. You don't have sorting protocol?
Yeah, for some reason, it's in the file, but it's not showing up here.
Here, try this. Oh, you know why? It's a continuous variable. Okay, never mind. So I'm going to start over. Well, not completely over. Go to, find your day on here. It should be here. Yeah, day. Select that column. I'm going to fix this. I love these quick fixes. So right click and go to properties. I think yours says double. Make it categorical. And we can make that a factor. Okay. Okay, everybody do that? Now go back to stats. Stats, ANOVA. Is it showing up now? Yay! Science! Okay, again, put in day, add factor, cell type, add factor. And then here's what we're going to do is we're going to hit day and then go down to cell type and hit shift. Select both of those. And then hit add interaction. Okay, we have our variables in here. Now what we want to do is we want to contrast things, right? This will only give me p-values. I want fold change information because I'm going to show you a program. When we finally get done with all this, I'm going to take these genes and put it in this program, and it's basically will tell me whether this gene goes up and down, what pathways might be activated or inhibited. So fold change information is huge for me. So here's what I want you to do is go to contrast. Okay. First thing I want is, instead of selecting day, go to day and cell type. Okay. I am interested in genes that are associated with differentiation that are different at before they get differentiated and then after they get differentiated. So I'm going to put this group in first, IPS cells in the top one. So I selected 0 star IPS cells. So that's a day. And I'm going to compare them to the ES cells. Hit add contrast. If you don't hit add contrast, it's not going to do it. Now I want to select, I want to see those genes that are different, that are also associated with differentiation at time, or the day 5. So I'm going to hit IPS cells day 5 for the top one. ES cells day 5 for the lower one. Add contrast. Now let's go back to day. I just want genes that change due to differentiated state regardless. So here's what we can do is we put day 5, this will be our differentiated cells, so add contrast. And then select the 0 and put it in the last one. Add contrast. And then let's do cell type. Since I put IPS cells on the top of all my other ones, I'm going to put the IPS cells here. Again, this is going to tell me those genes are those genes that change between the embryonic and pluripotent regardless of whether they get differentiated or not. So IPS cells at the top, ES cells at the bottom, add contrast. Okay, hit OK. Oh, and up here, data is already log transformed. Yes, it is. Remember, anytime you see all your values are between like 15 and like 1, that's log transformed. Usually log 2 transformed. Okay, I'm going to hit OK. Now here's what we're going to do is we're going to go to the advanced. So click on the advanced. This is an advanced bioinformatics class. I should do the advanced. Oh, there it is. What this is asking you is basically what I want to do is I want to see just the mean. Now select your day and cell type. I want the mean gene expression for each one of these groups here. I want what it is in the ES cells at time 0, the pluripotent at time 0, et cetera, et cetera. So go all the way down to all levels. Okay, hit add. Okay, I'm going to hit OK. I just have mean selected. LS mean, it's just going to give you the same number. But you can get anything you want out of here. I'm going to hit OK. All right. I think we got everything in. Hit OK. And I'm going to overwrite my existing file. Yes. You should, if you annotated your gene list right, you should see, once it's done with its calculations, you should see a bunch of gene names come up. Come on. Yeah, it's only doing 36,000 t-tests. I think it'd be quicker. All right, did everybody get gene lists? Got genes? No? Oh, did you? Oh, no. We'll take a look at it. Follow along. Remember, this is recorded. You can go back and do it. Yeah, that's kind of why I'm recording it, because, yeah, it's tough. Okay. Everybody else worked okay, right? Excellent. We now have all the genes and basically all these different, how different all these particular genes are at, whatever condition we want. Let's look at, and we could sort this by any way. So we can go to, say, day, p-value, and cell type. You can right-click on that and go sort ascending.